Hello, my name is Jonathan Jacobs. I'm the Director of Global Product Management for QIAGEN Bioinformatics. And today we're going to be talking about the analysis of human gut microbiomes and antibiotic resistomes with QIAGEN's Microbial Insights AR Database and the CLC Genomics Workbench. Our goals for today are first to show how to detect antibiotic resistance genes and their markers using multiple bioinformatics methods and databases, including the new QIAGEN Microbial Insights AR database, determine the taxonomic profile of shotgun metagenomic samples, build reproducible workflows for routine analysis of your data, and how to use the CLC Microbial Genomics module to identify potential mobile antimicrobial resistance markers found in plasmids and other mobile elements. And lastly, we'll visualize all of this data against reference genomes as well as against assemblies from your own data. So as you know, the issue of antimicrobial resistance is becoming a global crisis, and we've seen a global uptick in the number of resistant pathogens worldwide. And one of the best ways to characterize antimicrobial resistant bacterial pathogens is through the use of whole genome sequencing and well thought out and benchmarked bioinformatics methods. The CLC Genomics Workbench and the associated CLC Microbial Genomics module, which is produced by QIAGEN, has been identified as the top choice as far as commercial software is concerned for metagenomic solutions of clinical microbiology and public health lab data. With the CLC Genomics Workbench, you're able to analyze a wide variety of different data types beyond your microbial data, including human genomics data, other model system data, including RNA-seq or structural variants, variant calling. You can do routine analysis of your sequence data using tools like BLAST for primer design and cloning. You can do RNA structure prediction and map your data onto 3D models, as well as protein models as well. You can also do epigenetic data analysis, including ChIP-seq, bisulfate-seq, and other methods. And more importantly for this talk, you can use the CLC Genomics Workbench and associated microbial genomics module for a broad range of different applications in the microbial genomics space, including taxonomic profiling, metagenomic assembly, annotation and functional prediction of genes from metagenomics datasets, antimicrobial resistance and virulence factor detection and characterization. And if you're working with isolates, for example, in a public health setting or environmental surveillance setting, you can also create reproducible pipelines for assembly, gene prediction, comparative genomics, pathogen typing, including MLST, SNP-based typing, and Kamer-based trees, and as well AMR and virulence profiling. The QIAGEN Microbial Insight AR database is the latest addition to a growing ecosystem of capabilities within the microbial genomics module that includes a new database, new tools to characterize both isolate and metagenomic samples, as well as a growing knowledge base of commercially available data that was previously proprietary, which will be made available to the research community through our partnership with Ares Genetics. The new QIAGEN Microbial Insights AR database is an integration of 4 of the most popular publicly available databases, in particular, the Comprehensive Antimicrobial Resistance Database from McMaster University, also known as CARD, ResFinder, which is from DTU in Denmark, the Argonaut database, and the NIH's recently released AMR Finder database. We compiled each of these databases together, deduplicated the results, and reapplied a common ontology across all of the data making a single, fully integrated database for our users to analyze their NGS data with. We also provide direct links out to PubMed references for all of the entries within QMI AR so that you can kind of backtrack to the original source of why a particular marker or gene may have been included in QMI AR to begin with. Currently, the QMI AR database includes a little more than 3,800 unique peptide markers for analysis of shotgun metagenomic samples, as well as over 5,800 unique gene sequences for use with BLAST or DIAMOND to detect the presence of antimicrobial resistance genes in assemblies from metagenomics or isolates. We pair this database up with a series of tools, including a SNP-based point finding tool, which uses the point finder database, as well as an algorithm called Shortbread for analysis of unassembled reads from metagenomics data. And as I mentioned earlier, you also can leverage our nucleotide sequence database with BLAST or DIAMOND. All of these tools are workflow enabled, which will enable labs to process hundreds of samples in an identical way and promote the reproducible use of scientific research. This is a snapshot view of what you might see opening up the QMI AR peptide marker database. We include the resistance, phenotypic, and gene classification for every entry in the database, as well as live links, which if clicked in blue here, they would actually take you to EBI's website, and you would be able to explore additional details about the antibiotic resistance ontology data associated with each marker. We also include an attribution to the source database so that you can see where a marker originated from, as well as links to the original PubMed references that are supporting each of the entries into this database. Similarly, with our nucleotide database of gene sequences, we provide information on the associated classification of compounds that are the particular gene derives resistance against, and a phenotypic classification indication, as well as links, again, out to the antibiotic resistance ontology data on an external website. And briefly, the three algorithms that we provide for use with QMI AR include BLAST, Point Finder, and Shortbread. And these three algorithms expect different input, ranging from assembled contigs, whole genomes, or plasmid sequences, to raw unassembled reads obtained from either an isolate or a metagenomic sample. The output of these tools includes annotation of genes in your contig sequences and the binning of reads from the Shortbread output, so you can then further map them back to any reference sequences or assemblies that you may have to identify where a particular read that contains a marker might end up in your assembly. And lastly, with Point Finder, actually in the middle here, this also produces a gene variant table for SNP-based analysis and predicted amino acid change, as well as read depth and other coverage information that you might typically expect to see, for example, with oncology or oncogenomics type data. But we apply this in the context of bacterial resistance SNP typing. So for the purposes of this talk, we'll be reviewing the data from a recent publication from Marco Candella's group, where they analyzed the stool samples from 8 pediatric patients that underwent stem cell transplantation. Half of these patients developed acute graft versus host disease symptoms, and all the patients that developed AGVHD were characterized by an expansion of their gut resistome. Resistance genes extended, however, beyond the drug classifications that were used during the therapeutic course while these patients were hospitalized, and it opened the question of, what is the background resistome that these patients had, and would it be possible to predict adverse outcomes from stem cell transplantation therapy by looking at the gut microbiome of a patient prior to their procedure. So the data is all publicly available at the NCBI's Bioproject Archive at the link below, and it includes 32 whole genome, whole shotgun metagenomic samples from each of the 8 patients, as well as the associated metadata from the individual patients. And what we'll be doing today is building a workflow, which at first glance, this may look complicated, but we'll be stepping through each of the parts of this workflow individually. And the goal of this single workflow is to do both taxonomic profiling and AMR marker identification in a assembly-free context, and then also do de novo assembly, gene finding, annotation, and binning, which then will allow us to identify the specific contigs of sequences and the associated genes that have antimicrobial resistance markers associated with them. So in the first case regarding the detection of AMR markers directly from reads, the Shortbread algorithm was recently introduced into the CLC Microbial Genomics module last year. This algorithm is an open source algorithm that was published a few years ago, which allows for the creation of peptide marker sequences in a database, which are then used to rapidly screen NGS sequencing reads without any assembly. And reads that match a particular signature in the peptide marker database are separated out and can be classified as potentially containing a marker related to the database that was used. In the original Shortbread paper and in the context of how it's used in the CLC Genomics Workbench and Microbial Genomics module, we use the QMI AR database to develop a peptide signature marker database, which provides input into Shortbread, along with your own raw sequencing reads, of course. Running this workflow is very simple. It requires 3 steps. First, after importing your data into CLC, you would typically trim and apply a QC step to your data to remove any reads that are poor quality. And then you would run the Identify Resistance Markers by Shortbread tool. This tool then outputs a report that lists the antibiotic resistance genes of interest that were contained in your input data, as well as a binning or separation or partitioning, if you will, of your input FASTQ reads, so that you know which reads actually contain the markers that were detected. And it also produces a abundance table and associated visualization, similar to what you might find in a whole shotgun sequencing taxonomic profile. For those of you who have done 16S metagenomics or other shotgun metagenomics in the past, you may recognize this stacked histogram as being familiar, but instead of species level identifications, what we are looking at here is the relative abundance of different antibiotic resistance markers that are present in each of the 8 patient samples, each of the 8 patients across the 32 samples. And to the next slide. What we see here is that using this simple 2-step workflow, we were able to identify all of the same resistance markers that were identified by the authors in the original publication. What you see here in this figure is adapted from the original publication, but the data presented is actually data from using CLC Genomics Workbench with the associated microbial genomics module. And we've exported the data from our pipeline and into Excel, just simply to provide some additional visualizations and coloring of the results. We have the dates or the days post-operative that the patients were given antibiotic treatments, and you can see that in the red box we have patients that were AGVD HD positive, meaning that they had an acute response to the stem cell transplantation procedure, separated out from the patients that did not. And without any high-level clustering or other further sophisticated analyses, you can already see that there's a dramatic increase in the intensity of colors on the left-hand side of the table versus the right-hand side of the table, indicating a overall higher prevalence of antibiotic resistance genes in these 4 patients versus the other 4 patients that did not have an adverse outcome following the procedure. Furthermore, the data on the far left indicates the different resistance classifications associated with the individual markers identified by Shortbread. An important thing, a caveat to keep in mind, is that the Shortbread algorithm does not identify genes. It identifies marker sequences or peptide sequences that are contained within a gene. So an individual marker sequence may potentially be present in multiple genes if it was a resistance gene that was found in multiple species, for example. But nonetheless, when we transpose this data onto the data present in the original publication, we find a perfect concordance with our results here with those of the original authors. But resistance profiling alone is often not enough, and many times researchers would like to also know not just what the pathogens or bacterial pathogens are doing in a particular microbiome sample, but also who are they and what are they. And so that takes us to the second step of our pipeline, which is using a taxonomic profiling tool, which we've referred to as TaxPro for short, within the CLC Genomics Workbench. So the taxonomic profiling tool that we provide has been previously benchmarked as being a best-in-class solution for both 16S and shotgun metagenomics reads. For the purposes of this analysis, we did not optimize the underlying database. We just simply downloaded the default 16 gigabyte memory database from the CLC or the QIAGEN bioinformatics server through the software, and we ran a very simple 2-step taxonomic profiling pipeline on a typical laptop. And for reference, in case you're wondering, the typical amount of time to do this analysis on my laptop was about 15 to 20 minutes per sample. In some cases, it was a lot less. The depth of sequencing across these samples was not equal. Some of them had 2 or 3 times as many reads as others. But nonetheless, we've designed the default database to be able to run on a typical laptop. Now, we also have a larger database that's available, which mirrors the ProGenomes reference database. And users can also customize their own databases using the tools within CLC to fully characterize and maximize the level of resolution that they may need for individual projects. Nonetheless, for this initial analysis, again, it's a simple 2-step workflow where after importing your reads and doing some simple trimming and quality control of removing low-quality reads and so forth, we then apply the taxonomic profiling tool to produce a variety of different visualizations. In the lower right here, you can see a stacked bar chart. And in contrast to the previous data that you saw for the Shortbread AMR profiling, this chart is what you might expect. This is a list of all of the different family-level classifications of organisms that were present in the individual patient samples across time. Some of these patient samples had more sampling than others, but nonetheless, you can organize your data as you see fit. In the next slide, we see there are a number of other additional visualizations that are possible. And in the interest of time, we will not be going over all of them, but it's important to know that for the purposes of investigating potential AMR and bacterial pathogen presence in a microbiome sample, a lot of these tools are going to be key for individual clinicians or researchers to know how to use. So on the left here, you have a sunburst plot of all of the data aggregated for an individual patient. This particular patient had a very high level of Enterococcus present and detected in all of his samples. And indeed, this particular species that was present in his microbiome also was carrying a fairly large number of antibiotic resistance genes, which we can see on a later slide. This sunburst plot on the left here is fully interactive. You can drill down or back out as you need to explore the data at whatever resolution is appropriate for your work. In taking multiple samples, for example, the 32 samples that we analyzed with TaxPro, we then can do some principal component analysis. In this particular example, we aggregated our results at the family level. We applied a Bray-Curtis distance matrix and ran PCOA on these samples, and you can see that there is a very good separation of AG-VHD positive versus negative patient samples. The vast majority of the healthy samples are clustering in the center, whereas the adverse reaction and patient samples are sort of around the perimeter. Furthermore, if you wanted to investigate the level of diversity across your samples, which sometimes is important, for example, if you wanted to make a decision to resequence a particular sample or try to go deeper and get some additional trace level detections, you can visualize across your samples with simple box plots. In this case, for example, we're showing each of the individual patients, all 8 of them, and the individual samples collected from them, and the level of alpha diversity present in each of those samples. As you can see, they were fairly diverse. So, furthermore, we can take these same reads that we looked at before from Shortbread, bin them out, and actually run them as an antimicrobial resistance input data set. So in this case, we're still looking at species, or actually in this case, we're looking at family level taxonomic classification of the reads, but the input reads here, instead of being the total raw FASTQ file from each patient sample, it's just those reads that were identified by Shortbread as containing a AMR marker of some kind. And in this way, you can kind of see the relative prevalence of antimicrobial resistance markers identified by Shortbread across the various different patient samples. And this data actually correlates very well with the previous table I showed, or heat map I showed, where you could see different classifications of AMR genes in each of the patient samples as well, which is a different way of looking at the data. And what this gives you is the ability to look at not just narrow your focus not just down to the AMR reads in particular, but also to ask the question of which potential classifications of organisms are actually carrying these reads. So if we wanted to look further into the data, we would know which species to focus our attention on. So lastly, we are coming to the longer pipeline, which involves assembly, gene annotation, and binning of our assembled contigs, and then visualizing those results in various ways. This pipeline actually does take a bit longer to run. Altogether, it's about 40 minutes or so, 45 minutes or so on average, on a laptop, my laptop in particular, and for this data. And the pipeline proceeds by first trimming and QCing your reads, as we always do, using our de novo assembler that's included with the microbial genomics module, which is tuned specifically for shotgun metagenomic samples, by the way. We then, once we produce our contigs out of that assembly, we then proceed with gene finding and functional annotation of those genes that were identified using the QMI AR database, as well as followed up with 2 steps of binning. And for those of you who aren't familiar with binning, the idea here is that you have completed a de novo assembly and you want to ask the question, which species do these contigs belong to, or genuses or family level classifications can I make for the individual contigs? And if I can't accurately classify an individual contig, is there perhaps another way that I could classify them or bin them based on other sequencing characteristics like GC content, tetramer frequency, depth of coverage of the actual reads, etc. And so in our workflow, we apply both binning approaches in serial. First, binning out contigs that have a high level of similarity to reference genomes in our database. The default settings that we use are a 90% cutoff. So in other words, individual contigs need to be present in the taxonomic classification database at a 90% level of identity or higher. And then we also, because we use two databases, one for bacterial reference genomes, and we have a second database that we use for plasmids, which you can create your own custom databases as well and use them in whatever combination you would like. Any contigs that are not binned or classified using our taxonomic binning tool, which is called the Bin Pangenomes by Taxonomy tool, we then feed to the second tool, which is the Bin Pangenomes by Sequence tool. This then looks at those sequence characteristics that I mentioned before and further tries to bin them, bin the contigs appropriately and collect similar contigs together so they can be analyzed further. So just a quick visualization on the QC and trimming reports that you might see, and also perhaps some of the reports you might see from our de novo assembler tool. So as I've mentioned several times now, it's always important to make sure that you trim and QC your reads, and CLC includes all the typical reports that you might expect to see, including quality metrics of all of your reads across your whole data set, the so-called Fred score. And for the de novo assembly, we obviously report out the number of contigs, the average contig length, the N50 value, sort of their upper and lower bounds for high and low quality contigs within your assembly, and a myriad of other metrics, which are all available to be exported as PDFs, graphic images, or Excel files, CSVs if you like, etc. And later this year, with our next release, we'll also be including the ability to export all of the reports and data analysis from CLC in JSON format, which will be of particular interest to folks that are building high throughput pipelines. Furthermore, once you have an individual assembly done, you can dig into the individual results and navigate through all of your samples using our the CLC metadata tool. So within CLC, you can associate metadata with individual samples, starting at the individual raw reads. And as you conduct various levels of analysis and subsequent analysis on that individual sample, then the subsequent results and secondary files that get produced remain associated with that original input sample. So what you're looking at here is for patient number 15, a particular sample which was day 41 post-op, and the original file name or FASTQ run accession number is indicated on the far left of the table on the top. By opening up our metadata table view for this project and selecting that individual file, we're able to then find all of the associated data that has come from that file in the window below. And this creates a very easy way to navigate all of your results. Because once you start doing taxonomic profiling, AMR prediction, de novo assembly, etc., you might expect to find that you're going to have a large number of samples that you would need to keep track of. And this provides an easy way to do that. So focusing on this individual sample as an example, after we're done with our de novo assembly of that sample, we run our gene finding tool and our functional annotation tool, which we include three different tools. So the way that we analyzed the functional annotations of the assemblies for these samples was using Diamond, which is a very fast version of BLAST. It's not really the same as BLAST, but it's a close approximation of the way BLAST works. And the main advantage of the Diamond algorithm is that it's extremely fast, and you can provide it with a gene database of your own design and use it to annotate open reading frames, CDF sequences, et cetera, in a reference genome, or in this case, a de novo assembled genome in which the genes were predicted. So in our case, we've identified close to 11,000 predicted CDS regions within the assembly. We then did an annotation of that gene prediction with Diamond using the QMI AR database that was released in May. And out of that, it identified 711 genes with high similarity to known antimicrobial resistance genes that are present in the QMI AR database, or about 6.65% total. I'll also note that the underlying gene finding algorithm that was used here was our own internal tool, which is based on the Glimmer algorithm. However, within CLC, you also have the option to use the MetaGeneMark and GeneMark pipeline, which is a secondary commercially available add-on for CLC. So following the functional annotation of all the contigs within this particular patient sample, we then binned them. And in this case, this is a snapshot of the top of the PDF report that gets produced by the bin pangenomes by taxonomy tool. In this case, we were able to successfully bin 524 of the 1,400 or so contigs that were present in this assembly. And about half of those had a high level of contig purity, which is our measurement of similarity to reference genomes that are contained within the taxonomy database that you provide. In this case, again, it was the default 16 gigabase TaxPro database that we provide as an option to download. Two of those bins included contigs that had a high similarity to plasmid sequences. That was based on, because we also included our plasmid database. And there's further statistics available in the PDF from this taxonomy report. But more importantly, though, the individual contigs now have been labeled and annotated so that you know which bin they belong to. And if there's a particular organism or set of genes that are of interest, you now have a way to search through that list of genes and filter out individual sets of contigs that belong to a group of, to a taxonomic binning group, which you can then do further and downstream analysis on. For those contigs that are not successfully binned by the taxonomy tool, we then applied the bin pangenomes by sequence tool, which is based on a variation of Metadata and another algorithm that was put out by Salzburg Group some years ago. And this tool takes into consideration read depth, coverage, sequence characteristics such as GC content and tetramer frequency and so forth, and then makes a prediction as to how likely it is that multiple contigs within your data set actually belong in the same bin, independent of any reference database that you might provide. So after we are completed with our binning, we then can take our contigs and blast them using the Find Resistance with Nucleotide database, another tool that we provide within CLC. This takes contig data or individual gene data and blasts it against whichever nucleotide database you provide, which in this case we're providing QMI AR. We identified 23 different genes associated across this particular assembly for this particular sample. And these are genes that have been previously associated with resistance and have been annotated as such in our database. And again, you can see the table of results on the left here shows the BLAST percent identity score, the actual bit score, and the length of the hits, as well as the predicted phenotype, the compound class associated with that gene, and then there are live links that a user could click on that would take them out to the antibiotic resistance ontology site to get more information. But most importantly, this gives you an idea of which contigs have very highly qualified full gene sequences contained within them that could potentially be used as a lead to identify and characterize those organisms further in your data. Once we've completed our binning exercise, the next step that I like to do is take the original reads that were used for the assembly, as well as the reads that were separated out and partitioned out by Shortbread, and map them back to the assembly, and then visualize them within CLC in our track viewer. And this is a way where you can add arbitrary layers of annotation and other features, including point mutations and whatnot, and look at where the actual reads that are containing these individual markers might actually land in your assemblies. And what you're seeing here is a track view where we have an individual contig visualized across the top. We have predicted gene sequences, or CDSs, coding sequences, as the next row in yellow. We also have the total number of reads that were used to assemble this contig in the first place. And this is a fairly short contig of about maybe 15 KB or so, maybe 12 KB. And the individual Shortbread reads are then marked on the track below. And they're also further highlighted by this threshold track, which is a tool that we provide that allows you to quickly identify regions in an assembly that have reads that map above some threshold of interest. In this case, I just set the minimum threshold as a single read as a minimum. Is if you have a Shortbread read mapping somewhere within the assembly, I wanted to know and be able to visualize where that is and what genes were associated with that. And in this way, you can then roll up to the gene that is intersecting with where these reads are mapping and actually identify the specific gene that was found in this contig. And this provides you with an orthogonal way to identify potentially antibiotic resistance conferring genes that may have been missed by one of the other methods that we already presented earlier. So the nice thing is that all of these different capabilities and tools can be combined into a single workflow within CLC. And what you would do is build each of these workflows up separately, and then once you get them working, you combine them into a single large workflow to do a whole range of different analyses for you. And this view here, of course, is kind of a PowerPoint graphic figure of what this work looks like. But within CLC, this is a simplified view of what a workflow might look like. And it's very easy to build. These workflows take just about a few minutes to assemble. And it's a matter of just simply dragging and dropping individual tools and algorithms from the toolbar onto a canvas, and then attaching outputs of one algorithm into the inputs of another, and then configuring the underlying parameters of each of those algorithms if you decided to. Oftentimes, the default values for our algorithms suit the purposes of our users quite well, but you, of course, have full flexibility in tuning the individual parameters of each of these tools as needed. And so beyond the context of the case study that I presented, we also have the ability to present, to analyze point mutations. So we have a tool called Point Finder, which is able to analyze reads that are mapped, unassembled reads that are associated with an individual isolate. For example, if we have a database for Salmonella, there's another for Klebsiella, There's another for tuberculosis, MTB. And these databases include all of the SNP values, all of the SNPs and other variants that are associated with resistance, as well as the different compound classes and resistance phenotypes associated with those individual SNPs. The Point Finder database is maintained by DTU in Denmark, and we provide a direct download for our users within CLC to take advantage of this excellent work that group is doing there. And as a kind of an example of how this might be visualized, this is data obtained from a salmonella isolate that was part of a multi-drug resistant outbreak in Pakistan. And you can see reads here that were identified by the Point Finder tool, mapping with a high level of precision directly over the point mutation in the GYR gene that confers resistance to ciprofloxacin. Furthermore, using the CLC tools in tandem, it's easy to identify large chromosomal regions that harbor transposons and other mobile elements. And once these resistance genes cassettes are identified and you map your reads onto them, you can see, in this case, this is beta-lactamase. You can see, which genes are part of a particular resistance gene cassette, and then you can use that information for your for future experiments. And similarly, you can visualize these in a plasmid context and circularize your results. And the results you see here are from an analysis of predicted gene sequences found in a plasmid sequence. The individual gene annotations were obtained from a large database of peptide and gene marker sequences, and the functional assignments were made there. And then we can, again, use this data to then map our individual reads to see which of these genes are potentially highly prevalent in an individual sample, or you could even go one step further and use this data as part of an RNA-seq pipeline, for example, to identify which genes might be actually actively being expressed in an individual sample from a patient following treatment of antibiotics. So today we reviewed how to use the QIAGEN Microbial Insights AR Database and the CLC Genomics Workbench with the Microbial Genomics module to detect antimicrobial resistance genes and markers from clinical patient samples that were publicly available. We determined the taxonomic profile of these samples that were shotgun metagenomic samples. We also discussed how to build reproducible workflows for routine analysis. And we also learned a bit about how to identify plasmid sequences and also analyze mobile elements that might be present in your samples. And lastly, we also looked at a range of different visualizations on how you might want to see these markers in your data and compare them across multiple data sets long term. We also have an application note out, which is a PDF, which is available, where we go more into detail on this topic. In particular, this application note focuses on the Salmonella enterica multi-drug resistant pathogen outbreak from Pakistan that I referred to earlier. And if you visit QIAGENbioinformatics.com and look under the Microbial Genomics module resources page, you will find a link to this application note for you to download and read at your own leisure. Furthermore, if you have any questions, you can feel free to contact us by email or telephone. We have a one-day response time if you contact our support team. And we also have a large number of previously recorded webinars and other training materials available both on our website as well as on tv.QIAGENbioinformatics.com. And with that, I would like to thank you all for your time and attention. And please don't hesitate to ask questions and let us know how we can help you with your antibiotic resistance genomics work in the future.